Hello, it's a pleasure to be with you today, and it's my turn to present what are the levels that we see in ruminants. My role within dsm-firmenich is as marketing manager for ruminants globally, and I will be happy to walk you through the results that we see. This year, 2024, we processed around 1,600 samples of finished feed for ruminants around the world. This one comprises both concentrate feed, but mostly what we call TMR, or total mixed rations. So in most of the world, we see highest prevalence of the mycotoxins that we call the fusariotoxins, so produced by Fusarium. We see the most zearalenone, DON, and fumonisins. So starting with highest prevalence of zearalenone, you can see that there was a 73% of the samples that were contaminated by it. We also see 2/3 of the samples, or more than 2/3, were contaminated by DON and fumonisins. In terms of the concentrations that we see, we should be worried of the three, yes, but particularly the levels were going high for DON and fumonisins. On the other hand, aflatoxins, as you know, they're usually better controlled because they are known to be storage mycotoxins. However, the levels of prevalence that we saw the last year were relatively high compared to other years, with 30% of the samples, an average of positives up to 33, which is quite high. Okay, so why would you mind, especially focusing on DON and fumonisins, why would you mind those levels? The best way to share what this could be leading to is by looking into data that we have from 2020. In the Journal of Dairy Science, we published this research with the researcher Antonio Gallo, where we looked at two different treatments, so control and mycotoxins, the two treatments that I would like to show today. And we have a contamination level that is quite similar to what we saw in the rest of the world this year. So 825 parts per billion and 1,000 parts per billion for fumonisins. So with these levels of contamination during 3 weeks of exposure in high-yielding dairy cows in Italy, what this resulted on was a reduction of around 3% points on dry matter digestibility, which most of that was actually coming from the NDF digestibility, where we had a loss of 10% percentage point. This negative impact on the digestibility resulted in a reduction of milk yield of around 1.3 kilograms of milk. So this is what you could be expecting under these conditions if you were to measure and control the conditions like we did in the scientific trial. Why else would you worry, which is not the productive area part, but also the impact on the health of the animals? First, you should worry about the immune health of your animals because of the opening of the epithelial intestinal cells. So in this trial, we wanted to look at how much concentration of mycotoxins, especially DON, do you need to inactivate or damage 25% of the intestinal epithelial cells. Very important for the immunity of the animal, the diet digestibility, and so on. We found out that you need only 172 parts per billion. So if only a quarter or less than a quarter of this contamination that we see of DON in the average of the levels of the world would come into the intestine, we would already be having a 25% damage of the epithelial cells in the intestine. And lastly, we should also worry that if this DON passes through the epithelium and gets to the bloodstream, they should be having a negative impact on the immune cells of the animal that will be responsible to defend the animal against pathogens or diseases. You need only 62 parts per billion to kill or inactivate 28% of the immune cells of the animal. So an animal that is exposed to DON is an animal with a very depressed immune capacity. So with that, I leave you with the impacts on productivity and health, why you should be worried or cautious of the negative levels that we see in the contamination in the rest of the world. Thank you so much.